Good morning, everyone. My name is Beshoy Albert, and I am a sophomore student at the Biomedical Science Department, Zewail City. First, what is health? How can we define health? Health is the state of physical and mental well-being. But I'm not here to talk about health. I'm here to talk about the threats that face our health. We have a number of possible threats, including biological hazards, chemical hazards, and physical hazards. But I'm gonna specify or talk about the biological hazards. We know that bacterial infections are lethal and are the common cause of many diseases here. So a top pathogens that contribute to diseases are bacteria that almost everyone knows, such as Campylobacter, Salmonella, E. coli, and so on. But what could be a possible treatment? Anyone who would know what could be a possible treatment for bacterial infections? This is an obvious question, right? So the answer would be antibiotics, right? So how many of you tried antibiotics? Can you raise your hands, please? Okay, and when you tried antibiotics, did it work? And if it did the disease goes and if it was persisted? But if it didn't, why antibiotics didn't work? I'll answer that for you. It is antibacterial resistance or antimicrobial resistance. This is our monster here that we're talking about. So I need to show you how dangerous that antimicrobial resistance is. We need to show the Central Disease Control Report that released about September 17, 2013, that shows 2 million illness, 23,000 deaths, and 4 of 18 often transmitted through foods. That's very dangerous, right? So how did the media handle that? How did the people know about this? We have medias such as Scientific American, the BBC, and a lot of newspapers talking about antibiotic resistance. So is it really that serious? Yeah. And what if we continued using antibiotic resistance? What if we continued using this path? Unfortunately, it will lead to death. But why antibiotic resistance is really dangerous? This is a report by the WHO report that shows the first antibiotic resistance that was penicillin in about the 1920s, and the last antibiotic discovered was in 1980s. So are you really serious? The last antibiotic discovered was in the 1980s? Is that possible? Unfortunately, I've talked about the slide earlier, it will lead to death. So I'm here to talk about the amazing future, the hope, and the solution for antibiotic resistance. And the amazing future that I'm talking about is bacteriophage as an antimicrobials. So what is a bacteriophage in order to talk about bacteriophage therapy? A bacteriophage, from the word, it implies a bacterium means a bacteria and the phage means virus. So it's a virus that infects the bacterium. So what is the structure of that bacteriophage? A bacteriophage has a head that contains the genetic material of the virus, which is in this case the DNA or the RNA, a tail in which it injects the DNA into the bacterium, and a tail fibers in order to recognize the receptors on the cell surface of the bacterium. And these are the microscopic images of different phages. So why use phages compared to antibiotics? And the first and the most important therapeutic advantage is the specific target recognition. And I intended to use this picture here, the sniper, because it really implies and shows how important this feature is. We have self-replication and the phage, in which the phage enters the bacteria. Billions of phages are produced that keep entering the other bacteria, and the cycle goes on and on and on. Phage therapy applications are very, very wide. They're including improving food safety, industrial surface decontamination as an industrial port, animal therapy and growth, and of course, human therapy. And that's our hope. That's the promising future that we are waiting from phage therapy. So I need to talk about the phage life cycle first. The phage is, as we said, is a virus, but how it causes, or how it kills the bacterium. First, the phage, using the tail fibers, identifies the receptors on the surface of the bacterium. It injects its DNA through its tail fibers. It hijacks the machinery of the bacterium in order to synthesize its proteins and replicate its DNA in order to assemble these components into new phages that will eventually cause the lysis of the bacterium. And that is the lytic cycle of the bacteriophage, in which we are talking about here bacteriophage therapy. And in order to show you how phage production is really, really vast compared to bacterial growth, is 1.6 billion after 4th generation of the bacterium. So I'm here to talk about my own research at Zewail City University, and me and my colleagues have done this research at the university, and the aim of the research was to isolate new phages against the common pathogens such as E. coli and Salmonella for therapeutic agents. We first gathered the sample, isolated the sample in which the bacteriophage, we expect the bacteriophage to be present, in this case it was a sewage sample, so in order to target for E. coli. We incubated the sample with the bacterium in order to let the phage infect this bacterium. We centrifuge in order to separate the phage from the bacterium. We then performed spotting on bacterial lawns in order to detect the plaques that recognize the phage. We then performed purification to separate the phages from other phages, amplification, and then electron microscopy is our aim right now. And that is real research that done throughout the university. This beautiful plate here shows, this is our own plate, shows plaque forming the phages on the bacterial lawns. From the data, we have demonstrated CFU and PFU, and that are the basic of our results. The CFU is the colony forming unit, in which it is the number of the bacterium, while the PFU is the plaque forming unit, which is the number of phages on the petri dish. This is our own results during the university. First, the bacteriophage ZCAC1, Zewail City E. coli 1. As we show here, there are 3 lines. The first line is the blue line, which is the bacterial survivor. This is the numbers of the bacterium that survived after the infection with the phage. The green line here shows the control, which is the normal growth of the bacterium, and we can see a very, very huge difference, about difference of log 4, that is the reduction rate between the control and the bacterial survivor. The PFU here is, of course, is the plaque forming unit, which is the production of phage. That is another results using different phages on different strains of bacterium. But we can see here that this phage has completely destroyed the bacteria, at which it diminishes after 1 hour of infection, which is really, really remarkable, because that phage produced in the second results similar results, but with faster detection limit of the phage. So in order to discuss the results, we showed that bacterial survivor decreased, the plaque forming units increased, the phage production, and the colony forming unit, which is the control, increased as well. So as a conclusion, in order, because I'm really running out of time here, the conclusion is that bacteriophage therapy is really promising, and it is the hope of against antimicrobial resistance. And this is our photo with our beloved Dr. Ayman al-Shibbini at our laboratories, and we have won the best poster in the annual undergraduate conference at Zewail City. And thank you so much for this.